Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PIcc Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PIcc is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of the discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward. Now.
Wonderful, wonderful. So welcome everyone to the steering committee. We're here with the Pathology Innovation Collaborative Community, aka Alliance, July 28, 2021, Earth. I hope you're all doing well. It's a little bit of a slower agenda, so we should have some time to do some additional discussion. Let me just share my screen and start with the updated agenda that I think Ulla just pasted here. So, can you guys see my screen?
Yep.
Right. First of all, some opening remarks that I was asked to repeat, which is we follow our code of conduct, the official charter and the conflict of interest policy, which you can find here, as well as our rules of engagement, which is summarized in a beautiful short video that's synchronized by a friend from the UK on YouTube. So please refer to that for all of our sort of rules and engagement. For those of you who are new here, I thought of briefly using a minute to explain what this collaborative community does and what this looks like practically. So if you value collaborations, you're at the right spot. You may have an idea for a regulatory science project. You can present this here just by submitting your project idea or concept via email to Ulla, who you see on the screen, and maybe you can provide the email. It's also all over the website. You propose your project and you can present this or send us slides and we present it for you at one of the steering committees or one of our other project meetings. And then this collaborative community helps you organize the project. Thereby, you have or tap into a network to find other interested collaborators that would like to do some regulatory science. First of all, the focus is not to create a competitive product. We're in a pre-competitive space. And the second part is that PIcc as an organization is not actively participating in terms of like shaping or redirecting your project. That is all with you. So in essence, we provide an infrastructure to connect all kinds of stakeholders that you find or may find interesting or relevant. As such, there's about 400 people on a website and on our mailing and distribution list, and all the information that's shared here is made publicly available, unless you want to do some editing, but that is sort of the concept. And I thought it may be helpful to reiterate that, especially since we have a few new people on the call. So let's start by going through some of the projects. I think you have the agenda in front of you. The first part was the request to put a couple of the interesting ethical or ethic and governance papers about AI on one subpage. So this is not directly a project, but it's intended to be the go-to place for pertinent literature. If you see anything from the radiology space or from, your personal experience or just stumbling over something in PubMed, please feel free to email that. Again, here's our email, and we can put it on this page. And it's, again, intended just to be sort of an online resource. Questions, suggestions about that? If you have some additional ideas about this topic here, I think it's just a good place to have the relevant papers. I think the next update is from the high throughput truthing group. I don't know, Katie, Brandon. I took a screenshot here. If you want to share your own screen or other stuff, then feel free to. But
No, actually, Kate, maybe you can put a link to this page. It looks really familiar to me because I just shared it with my digital pathology colleagues here at the FDA. Trying to harmonize all these updates that I end up giving so that I don't have to do it as often, but as comprehensive. But just so you know what's happening in our project, we're trying to get real serious about the pivotal study and defining the patient population therein. This is tumor infiltrating lymphocytes in breast cancer. So that's what we're going to be discussing on the 6th in our project meeting. And then at the top, I've got a little bit of information about how to get more information about the project. And then coming up on the 8th, it's a Sunday, is I am presenting at the Joint Statistical Meetings. Some of you may have encountered my pathology informatics talk. And basically, all the figures that were created therein and the data are going to be part of a GitHub R package. And that's going to be going public in the next, probably, I'm hoping right in parallel with this meeting. That the purpose is to share some of the statistical methods that we've started to explore, share the data, and get some statisticians interested and involved in this project as well. Shortly thereafter is the FDA Annual Summer Student Science Poster Day. These students have only been around like a month for summer, but we already got a couple posters. They are, so it's not some real high-level science, but kind of talking about what they're working on, a quick update, and how they fit into the whole project. And so, interesting, a great opportunity for them to showcase and learn about actually presenting science. And then we are actually recruiting right now, probably to take action in the next week or two at Yale. We hope to also send a EDAP system to Stony Brook, but if you turn the page, we have shipped a microscope system to Yale, and they're putting it together. Sarah is leading that. Sarah is leading that data collection effort at Yale, and so here's some, a little bit further down is information about the EDAP system and about including who we got. So yeah, there's a link there about recruitment information in case you can share it with your networks. I might try and pull some strings as well to share those recruiting documents elsewhere. So you might see it again. Sorry for any spam like that. Scrolling down, you can see that CWEN presented last week, actually 2 weeks ago, on some other statistical methods related to this data. We're going to be posting the video from her talk, and she also already has a manuscript submitted for almost 6 months now. It's taking forever to get published, so we decided to put it on archive. This is going to be ANOVA methods for multiple reader, multiple case quantitative data analysis. The paper is a little bit statistical versus the presentation. She started to apply it to the high throughput truthing data. And so we're going to get her slides and a video of her talk online. And a big milestone for me, many years actually in the making, but 6 months from submitting it to the FDA Council, we have 2 research collaboration agreements executed, signed by the center director and everything. You can see an abstract of those RCAs there, and the prelim documents. These are what I use to start the process at the FDA. So that's really exciting to know that Emory and Stony Brook are in it for the long haul and will be looking to do the slide sourcing. That's why the patient information was so important to get that figured out so that we can decide on the metadata, the covariates that we will collect along with the images, make it the appropriate patient population. And then you can see I've got 5 summer students. There's actually 2 more, but they're not actual FDA staff yet. And really working on communications and documenting that our package I mentioned, and increasing the features and functions of our data collection tools. So, and as well as collecting data. Sarah is back on the FDA payroll to do this at Yale, and everybody here should know her already. She's been a key element of PIcc since its beginning. And 2 of them are high schoolers, and you would not be, the posters that you will see that I mentioned above are done by high schoolers. One of them is done by high schoolers, the other is done by an undergrad. Really exceptional integration of young talent to develop over time. So thanks for the chance, Joe.
Perfect. Thanks a lot. Speaking of trainees, maybe we can turn it over to Sarah and Julia for a brief update on the trainees group.
Sure. We just have one update where we want to introduce our next keynote speaker. The tentative date is going to be in October. We're going to announce that through the website and Twitter. It's going to be Siba Al-Hussein, who just recently transitioned to the University of Rochester in New York, and is going to talk about the importance of AI for trainees, specifically useful online resources to understand machine learning better, and refer to coding classes, and just in general how to initiate new projects during your clinical training. So we're very excited for her and her session, and there's going to be a specific date soon.
Awesome. Thank you very much for pushing the trainees along. Laura Lassiter, hi, good seeing you. You have an update for us?
Yeah, I just wanted to intro, and I'm sorry, Abigail, I didn't give you any notice on that. But I just wanted to introduce, since I've transitioned over to pharma, but staying involved in the Alliance, and my amazing colleague, Abigail, is, I invited her to join. So I just wanted everybody to know Abigail is here and is going to bring a lot of really helpful pharma perspective to the conversation. Please reach out and say hello to her if you get a chance.
Awesome. Thank you very much, and great having you, Abigail. Let me just continue here. The next update, I don't know if Keith was able to make it. I don't see him on screen. Keith, are you there? In any case, so this is about a publication that came out as part of a series of 6 articles. And as you can see, this got quite some hits. This came out yesterday. The link is here. Maybe, Ulla, if you can paste it to the chat, that would be great. Briefly, this is about multiplex immunohistochemistry and immunofluorescence. It was a series in the Frontiers in Molecular Biosciences, and this is the one paper that I wanted to briefly feature. Without mentioning a lot, there's 9 noteworthy conjectures in there that should illustrate what is important regarding tissue multiplex analyte detection and anatomic pathology. And as one of the things you see here, Kim Blenman contributed, and aside from the authors here, there's an acknowledgement for the Alliance/PIcc group in here, which is great. So this is another publication where some of you have contributed, so you may find some of your ideas from some of the sessions back in here. I just wanted to point this out as part of this series. I know several of you are interested in, multicolor immunofluorescence. So this is like a, let me just pull this up real quick. It's like a full series. Oh, this is the link to the paper. I provide that in the website update, but this series is quite impressive with 6 different papers from various groups. One element that came up several times was, can we use certain standardized slides for doing some QA or QC work with whole slide imaging? And in our discussions with MDIC, one element that came up is that there is a so-called surrogate sample framework existing under the MDIC umbrella. This is the resource. You can also just Google surrogate sample framework. It leads you directly to the MDIC website, and you can download a document that was created in 2017. And as part of that document, for assessing IVD performance, for example, in the absence of a gold standard, what MDIC and a whole consortium of people put together is a very well composed document that contains several of the things that we may care about, especially when it comes to creation of artificial samples and artificial data. And I wanted to point this out briefly because there were several emails about, sort of a rubric creation for, standards of color reproducibility, etc. So I think I wanted to point out that this surrogate sample working group under MDIC has tackled this, including sample hierarchies and approaches to standardize this. And I just wanted to point out that these 2 components here, of course, may relate to some of the MDDT programs. So I just wanted to point this out. Questions about that? Or I don't know if some of the people who emailed about sort of surrogate samples for whole slide imaging, or if there are some ideas by the group. No? Yes? All right. Let's move on. After several conversations sort of offline, 3 people agreed to join the so-called privacy pilot project, or if you call this here as an abbreviation, feel free to do this. So we are still looking for folks to join, for help. And the reason and aims of this were several recent regulatory or legal developments. So 21st Century Cures Act, some of the information blocking rules. There's a whole HHS-driven approach to change HIPAA. The Department of Health and Human Services Privacy Program has recently put out some changes. There are a lot of state-level changes, and obviously there's one big issue between Europe and the US is that the GDPR framework versus the HIPAA framework are covering some similarities, but is quite different. And this alignment, this was sort of the first part of a longer conversation about what this group should focus on. And I put together sort of an outline comparing GDPR versus HIPAA at the very top level with certain privacy requirements under HIPAA, but not necessarily under GDPR and vice versa. What you see here in this tiny little inset here, and the PowerPoint presentation will be available for review on the website, is that certain states have different kinds of individual legislations already in various stages, and that there are additional approaches to govern privacy and or data security via, for example, certain conformance statements or certain certificates like the SOC 2 initiative. So this will again serve as sort of an outline regarding some of the privacy concerns. And one of the key things, I don't know if Josie and Trappin is on, but certain states have already relatively tough guidance on what PHI data can be shared across state lines. So this has implications for probably clinical, for industry, for research, and probably the broader realm, not necessarily only pathology, but this was sort of an outline trying to account for some of these issues. So this is only one of the levels that triggered this entire privacy pilot project. But I wanted to share this and ask for input, solicit additional members, folks, concerns, comments, etc.
Joe, maybe you could explain what the pilot is.
So the pilot is that we don't have a concrete aim or task regarding how to tackle this from a regulatory science perspective. So the pilot is just to solicit sort of the documents and compare the relevant documents and understand what is intended to happen. So for example, if a company right now tries to do data sharing across the Atlantic, basically they face the issue of GDPR. They can read that. That's a multiple 100-page document. But the key concerns for, let's say, sharing of research data follows rather simplistic paradigms, not necessarily the full rule set of GDPR. So one idea was, could we put together in a pilot sort of the key salient points so that researchers can share data? That was one of the concrete elements. But there are many, many other components that are relevant for certain laboratories. For example, if you want to share whole slide imaging data and metadata across state boundaries, some of these legislations here are already prohibitive. So now we basically see that the state level legislation diversifies from the general HIPAA framework because Consumer Protection Acts and privacy law is not quite the same across different jurisdictions anymore. So one other idea was, well, could we propose, for certain applications, certain guidances or what should be in there? And then obviously the big question is, well, is there any evidence that governing privacy in such a restrictive fashion has actually true benefits? That's more of a scientific approach. So for now, we've been just discussing this, so there hasn't been a concrete sort of aim in terms of like, should we write up a paper? Should we put together sort of just a web page as a resource? So we haven't really solidified how we're going to tackle this. I think, the ADAS group has put out a survey about information blocking. That was one of the big things that we discussed as well. So long-winded answer to saying we haven't really concretely defined what the pilot is, but we want to, focus on some of the issues that is most appealing to the membership.
Okay, thanks. That's helpful. I can imagine that a lot of the real world, electronic health vendor organizations are thinking about this as well.
Yeah, I think, multinational trials are definitely affected by this. Data sharing between Europe and the US is affected by this. Simple solutions for anonymization, for example, of whole slide imaging files are not currently easily available. It resides either with the PI and or just obtaining it primarily anonymous. I don't know, Brandon, how you guys did this with the HTT project, but it is definitely of concern. And some institutions we know take the whole data and strip them of identifiers and basically create duplicate research archives, but that gets very costly very quickly. So this will become probably more relevant the larger your data sets are and the more versatile you want to act with those. So
Yeah, Joe, I was thinking about the aim of a training program, is the first thing that comes to mind on this topic, because most people in this space that are developing algorithms or the like, might have no clue or might have some clue, but to have a fact sheet even, or just a training for that kind of for different audiences maybe, but I'm throwing too much at you that when I say that. But these slides, I'm sure they get into some of those details, but really focusing on the dangerous zones, the things that people need to be careful with is, I know from my desk would be nice.
Yeah, so I think this mini 17 slide deck was used to kind of run it by some of the folks at NIH, just to raise awareness that GDPR happened, actually a few years ago, 2018. But in the US, the data exchange is primarily affected, it seems now, because the privacy law discussions are happening right now. To those active in the privacy law field, this is also old news, but it's affecting a lot of things right now. So for example, HHS in this sort of privacy program has realized that a lot of the COVID-19 issues are raised, for example, reporting, contact tracing, etc. So that touches upon some very sensitive privacy components. So HHS has looked into, hey, how do other countries do this? And GDPR came up. So there's a big push to, revise HIPAA, so that may come. But I agree, sort of a fact sheet or, putting the salient points together that people know what is a SOC 2 certificate, because that comes on quite a bit when it gets to audit trails for servers, or when companies get into, I don't know, a product gets into, let's say, an academic medical center. We always fight with IT. Is this all appropriate? Can this be inside the firewall, or does it have to be secure on a local network? So it touches upon many different things. So anyone interested, please, shoot Ola an email, and we definitely have a follow-up meeting about making it a little more concrete. But this may become to some a hurdle, to other a field to become an expert in, to really maneuver things forward in a more sophisticated and efficient way. Other questions about the privacy pilot? So if not, we move on. Not sure if you've all seen this, but the Valid Act, in this case of 2021, was recently reintroduced. This is a slightly modified, revised version of the prior two versions. They're both Valid Act of 2020. What this is sort of a revision or a proposal to revise the oversight over IVCTs, which is the in vitro clinical tests. And the whole act, when spelled out, is Verifying Accurate Leading Edge IVCT Development Act. So the emphasis here is really on development, because it is covering basically what has been previously called laboratory developed test, specifically high complexity testing, by introducing a completely new framework on how to assess the development, planning that, the design, modification, and adoption for various intended use cases. So the intended use meaning what, and the indication of use meaning who and why, including the instructions of use, which is how, has not been traditionally under the purview of the FDA. And so for the first time, some of these tests mention, I put in what is encompassed here in sort of 9 key elements. And what is basically being proposed is, this is a 245-page document that is trying to revise a specific part, specifically section 201, that should be amended by this 245-page document. And there's a lot of sort of like definitions here that may sound familiar to some, but this revised oversight over the development of IVCTs will affect a lot of laboratories, and I think is highly pertinent to the field of AI, overlap with software as a medical device, but also IVCTs when it comes to, for example, diagnosing cancer, because that would likely, when using various devices, tick off the high complexity component. I know some of you have probably started reading this. It's quite an intense piece of literature. I don't know if there's other comments. I know, Shannon, you've been quite involved with this, so I don't know. You have some comments on this?
Yeah, I've read through it a couple times. It's super fun. I highly encourage everyone to do it. So basically, not only does this move LDTs into regulation by FDA, it creates a whole new class of product, an in vitro clinical test. And what that does is it moves in vitro diagnostics and LDTs under the same regulatory framework under FDA. And there is a section in the definition of an IVCT that includes software. So this could also potentially include AI, ML algorithms.
Are there
Yes.
any substantial changes from the previous iteration?
So from the 2020 version, it is 95% the same. They made some tweaks for the EUA section, and they made some good changes to the technology certification process, but otherwise it is largely the same.
It's a hefty piece that will require a lot of guidelines and additional explanation to really make it useful. So I think this is, obviously it is up for discussion, but there's many components of this that could be tackled from a regulatory science perspective to really, for example, align what the intent is with what it is proposing to accomplish and whether there's any scientific data or basis for that. So I thought that, if there's interest, we could discuss this. It would, however, require that you familiarize yourself at least a little bit with, for example, the text certification approach that is portrayed in there, or how an IVCT would be defined. So we will share the text that's publicly available. So if you Google it, just make sure that you get the valid act of 2021, not one of the two prior versions. But yeah. So would there be some interest in putting some of the components, together in a more maybe legible and easier format, akin to what, Brandon suggested, so that we could have like a mini fact sheet on the valid act and how that's applicable maybe to pathology?
I think it's a good idea. I think that it's highly relevant, but few people in the field have the time to really go through it.
Yeah, so the key element here is, again, we have to be just careful because this is legislation. So if we comment on this, we can only work on it from an educational perspective, not from a health policy perspective. But I think there are a lot of experts on the call here that will be able to help walk this nuanced fine line. But the key element that I think is very important is that this is happening right now. A lot of the laboratory directors in the various AP and CP programs are probably not aware what this will entail and what this will mean for laboratory developed tests. So putting together sort of a mini overview that gets the core facts out may be very helpful. So I don't know if Friends of Cancer Research or MDIC will join in on, creating sort of a brief summary or so, but I think that may be very helpful to have.
So the one caveat I'll add is, when you're reading through like the submission requirements for if one needs to make a submission to FDA, there's a very strong clinical chemistry feel to it, or even, next gen sequencing feel to it, like a wet bench type test. I don't know how well the language as written would apply to a piece of software like, an AI algorithm for digital pathology purposes. So that might be an exercise to walk through, is taking a use case or a couple of use cases and walking through the submission process to see if you're dealing with strictly a piece of software, are there any elements of the process that maybe don't make sense or would need interpretation?
When I read it, I was thinking about PDL1, right? Just an IHC stain. It by itself, an IHC stain would probably not classify as high complexity, but when directly tied to a drug that can have severe side effects, I think that definitely classifies. So I like the idea of trying to, run through it as sort of like a trial run. That sounds great.
I second that.
Okay. Let's move on. As some of you may know, the IVDR, which is the in vitro diagnostic regulation framework in the US, in Europe, has been passed, and it will affect roughly 25 nations and 450 million people. So I think when adding that together, one would consider that a fundamental change. So there are tons of fundamental changes happening. And there's a fantastic overview document. I don't know, Ulla, if you can paste in the chat right now, that was put together as sort of, I don't know, a so-called ECL submission from the Association of the European Cancer Leagues. So this is something like ECMO, but a larger group of leagues focused mostly on cancer. But the regulatory outline and summary of what this will mean for regulatory science is phenomenal. It's a great visual aid, and it's perfectly explaining how to align the intent with the consequences. So this is obviously a work by the EMA, the European version of the FDA, and they have put out clear-cut regulatory science goals. So if you haven't seen the document, I strongly recommend taking a look. It's quite busy. There's a lot of graphs and how they think about, for example, creating some of the real world evidence, or how to streamline data across nations or across states or across settings is quite impressive. So I just wanted to make that as a brief pointer. Question is, I think there is probably a new regulatory science strategic plan coming from the FDA. I don't know. It hasn't been published in a while. I think the last one sort of ended last year. I don't know, Brandon, if you recall, but or if you know if something will come up soon, but this is kind of like outlining, at least for the next couple of years, what the EMA is thinking, and this particular document is condensing this down.
Hey, Joe, you know, those are the things that I wouldn't be able to say, and even the whole Valid Act discussion, I probably would not even tiptoe into that. It just would get me into trouble.
Understood. Yet highly relevant to the field.
Well, if you want to think about data to collect that is related to that, that's where I would come in.
Absolutely. Speaking of, other documents that we wanted to point at, this came out, I think, 3 days ago. This came from the U.S. Surgeon General's Advisory Committee, and the U.S. Surgeon General gave a relatively long explanation for a document that's only 17 pages, but it is highly informative, and it tackles health misinformation. This was a direct consequence to some of the COVID rumors and weird articles. It includes what people can do in terms of taking action. So it covers quite a few ranges here. You see whatever your favorite branch is, where you would say where you see yourself. So it's quite a good document with only 1 or 1.5 page summaries of what individual, let's say, health professionals or technology platform developers can do to prevent medical or health related misinformation. This may not be directly relevant, but some of the outlines that are portrayed in this document clearly hint at that, of course, with the appropriate evidence, ideally scientific evidence, you can prevent misinformation. So a lot of the components, the wording, and the aims that are portrayed in here fall in line with what regulatory science is trying to accomplish. So in case you're working on a regulatory science project and you're looking for appropriate ways to word this, I would definitely recommend this, because it clearly outlines from at least 10 different vantage points exactly what is needed to prevent medical misinformation. And through the grapevine, I heard that there will also be NIH grants becoming attached to this to tackle really prevention or elimination of medical misinformation. So there's gap analyses written in there. Obviously there's the technology platform issue is tackling some of the AI topics that we discussed. So just wanted to point to it. It's a much easier read than the Valid Act, in case you can only read one document, just obviously a joke. All right. let's move on. I think next is an update from Kingsley Ibari from the Standardization of Data for Pathology Research Group. Kingsley, you there?
And
Hi,
Joe, I
Joe.
have
Good
some
afternoon.
slides I'll share for Kingsley.
Thanks, Sula. So I'll just give a brief rundown of what the committee has been up to since the last meeting. Can we go to the next slide? Next two slides, actually. Okay, so this committee is interested in providing standardized pathology data for research in these three levels of standards, including knowledge representation, DICOM, and then terminology standards like SNOMED. If we go to the next slide. So the basic problem we're trying to solve is how can pathology data be utilized and streamlined for research purposes efficiently and for FDA submission? And how do we create structured and standardized data sets from the LIS for pathology research purpose? The next slide. So the next slide shows that, in the workflow of pathology, when we get cases, anatomic pathology now, we never think about research. It's usually at the, as an afterthought. So if we go to the next slide. So we want to bring pathology research into view when we create this workflow for pathologists. But how do we do that? We want to develop, first of all, a model data standards, end-to-end data flow, and to provide a general framework for organizing anatomic pathology information. We want to also create a program that can extract and transform pathology data into standards that we need for pathology research. And obviously, all these processes will be to shorten the turnaround time for research projects. Okay, next slide. There has been a similar initiative that has been done before. The FDA worked with UCSF and Quantum Leap Healthcare Collaborative, and they were able to map data from EHR into EDC for clinical trials, and using specific industry standards like CDIS. Okay, next slide. So this was called the One Source Project, and it was a collaboration between FDA and, like I said, and with UCSF along with Stanford University, and they were able to at least give a deliverable for phase 1 of this project. That's what I think is available online. Can we go to the next slide? They used these different data standards, which we are all familiar with: HL7, IHE, CDISC, and therapeutic area data standards, in creating standardized data sets from the EHR to be used immediately for pathology research. And next slide. So the action plan for us is that we want to create our action plan is going to be in 3 phases. So phase 1 will be to create standardized formats of pathology data. And then CDICS, we create programming also to map this framework and to convert these data elements into CDICS standards. Our basic raw material or tools that we'll use will be the CAP protocol. And then we should be able to, at the end of phase 1, provide a standardized data set, an XML file that explains everything, and a data dictionary, and as well as an implementation guide for any person who wants to use that. And then phase 2 will be to incorporate digital pathology data into phase 1, and then phase 3 will be to incorporate biomarker data into that. For those who have been following this project, my presentation previously, phase 2 and 3 were kind of different, but now we want to just focus on this, create goals that we can accomplish. So this presentation was done to CAP members, the CAP Informatics group, with Samantha Spencer, Mary Kennedy, and several other CAP Informatics group. They were I think there are areas that we need to learn from how the CAP protocols are being stored and how they are being generated. And I think they'll be making further presentations on that on the 6th of August. So we are going to look at that, as well as looking for a way to use that knowledge of that to create, to achieve the goals for this project. So if we go to the next slide, I'm almost done. So our use case will be, for phase 1, will be to look at breast lobular carcinoma neoadjuvant post-therapy resection of pathology data, and to see how well we can map this data from available data elements stored in the LIS, and then to see how we can explore this using our data structure that we want to use. As for this, I've had some discussions with Dr. Edgerton of MD Anderson, and she's going to be working with us on this project to see that we, within the confines of what we can do, to produce something that will be beneficial for this group and for all pathologists. Next slide. So basically, this is the workflow that we hope to achieve for phase 1, and then we're going to work towards that. We are going to submit an IRB for the study by next week. Hopefully, we've made progress from the last presentation, and we might even have to look at some data elements from the CAP protocol. That's the back end after next week's presentation. I think that's it. Can you go to the next slide? I think this is just to say the level of conformance for the different levels at the end of this project. And then if you go to the next slide, it's just to thank everybody that has encouraged us to work on this project. Let's see how we can push forward. By next week, next month, we should have a tangible project progress report to give for this project. Thank you very much.
Thanks a lot, Kingsley. I'm sure there are many questions, because this is touching upon some of the core elements of what we're doing, at least on the AP side. So maybe I open it up for a few minutes of questions to the group.
I'll go ahead and you go first. All right, I'll go first then. So this is definitely ringing to the stage of my project where we have the research collaboration agreements done, and we are mapping out the metadata we want for our samples, and then based on those and our sampling scheme, we will, pull the specimens and prepare related slides for our pivotal study. So I just want to say that out loud. I'm fully want our work to either inform standards and interoperability efforts, and certainly maybe there's something that we could learn that if you were part of those conversations. And I think while I don't think day to day getting into the weeds of it, but at some point to connect on our plans and even just the operational steps that we're going to do. Now, we're not talking about starting with the clinical, putting this in the clinical record, but we are talking about extracting a lot of information from the clinical record in order to sample the patient. I hope we can connect on these issues.
Yeah, go ahead.
I'll let you answer and then I'll, sorry about that.
Okay, so yes, definitely. We are very open to ideas, collaborations. At this point, really, we're taking baby steps because we know it's a very complex process, and the more help we can get, the better for us at this point. So definitely, I'm sure we're going to learn a lot from just speaking with you. On what you're doing and sharing ideas of what we want to do. As much as possible, we want to keep phase one as simple as possible, so that the deliverables will be achievable at the end of the day. We'll not spend years, months, trying to get to phase one. So
I think similarly, we're going to do our slide sourcing and sampling, but the more we at least connect at some points, the more you can see the issues that come in a project that's rolling along, and the more we hear them, we can maybe take some advice that you, I'm sure, have to give as well. So
yeah,
I'll try and keep you in mind as we have some of these conversations.
Thank you so much, Brandon.
So Kingsley, I was surprised about that this was a UCSF initiative. Ours, although we use CAP Synoptics, it's pretty much a home brewed, and it's not, and we have very subspecialized faculty, but it's not like a drop down type of data. It really is, faculty put in whatever type of data they want, and then it goes to the cancer center, to the cancer registry. So I'm just curious, as you're doing this type of work, I'm sure there are individuals who really have structured data, but ours is really a natural language. So I was surprised that it got converted to some sort of structured data that's usable widely. And so I'm curious about, if you're doing this with multiple institutions, how you're going to separate all of those out between truly structured data of a specific diagnosis and how people write things that are completely unstructured. So that's why I was just curious about.
Yes, in fact, that's the idea behind this project, such that we could do multi-institutional studies, combining data elements from different institutions. We want to start with one institution as much as possible to get what they are doing currently, and to capture the data element as is, and then convert these data elements into structured formats. In fact, having different institutions with different data structures or elements speaks to the need for such a project like this, because we can create formats or programs that would, at the end of the day, standardize the data, such that if you want to do a multi-institutional study, it's maybe a click away, as much as possible. So I agree that the programs might be tweaked at the end of the day. It may not be a one-size-fits-all process, but we feel that it would be easily converted, because the CAP protocols, they're not exactly the same, even compared, if you compare MD Anderson, for instance, to Baylor, of which I had experiences from this institution. The CAP protocol as they are using currently is not the same, but they are very similar. So not so far apart, but we can get there if we want, with some good level of planning and thinking. And so that's where we are now.
Thank you.
I'm just gonna offer, Kingsley. It sounds like there's a similar thing done for EHR data with the Odyssey community. I'm not sure if you're familiar with OMOP and Odyssey. They just might be a good resource to draw inspiration from in their mapping and standardized mapping of EHR data from the institutions.
Yeah, I kind of looked at the Odyssey data before, but now that you're saying it, I think I'll take a deep dive into what they did and how we can borrow from that to create what we want.
Niels, you had a comment too, or?
Yeah, hi, sorry. I'm actually driving someone to a COVID test right now, but now we're in line. Anyway,
I hope it's FDA approved.
it is FDA approved. We're at Stanford. So anyway, the point I was trying to make is I've had really good success doing subclass labeling. So, you know, a machine trained at one level of an anatomic pathology problem, and then you really want to understand where it fails. So you have to label the subclasses that are inside that. I've had really good success with the SNOMED CT code. So for image analysis problems, I guess I would say that seems like the best starting point I've found, and certainly that's baked into anatomic pathology reporting already. There is probably some work to do there for features that are not yet put in there in SNOMED. For example, like they have germinal centers, but they don't have marginal zone, light zone, dark zone, mantle zone, all that kind of stuff. So I guess one thing I think about in my head is some version of what you're going to emit as a product, like a annex to SNOMED CT, question mark, over?
Yes. In fact, that's why at the beginning I showed the intersection between terminology dictionaries like SNOMED and the data elements that we get from the synoptic reports and digital pathology. So what we would like to do is to see how we part of the, I think, CAP group that has been listening in on my presentations. I think I've presented twice to the CAP group. There's very many SNOMED experts there, and we plan to use SNOMED CT as a dictionary level reference in the data itself, such that we can easily map data and transfer data from one institution to the other.
Awesome. And if you're around, you know, I'll look for your email address, and since I'm nearby, I might come by and say hi.
That would be awesome. I would just place my email address in the chat.
Fantastic. This is a collaborative community at work. I love it. All right. We have two more short items that I wanted to briefly touch upon. One is that we invited, hope we haven't heard back yet, from this group here. Despite all the numerous papers illustrating that AI can solve all our problems, this was a paper that says that computer-aided diagnostic systems for colorectal cancer from whole slide images are not quite ready. And I thought that getting a story like that in right now, including that they offer the whole CRC whole slide imaging data set available upon reasonable request, made this a very interesting group. I don't know if some of you know some of the folks here to help solicit that, but I thought it would be great to have them present a talk to PIcc and the Alliance. And, you know, Scientific Reports is not a bad journal, so I just wanted to point this out. The other thing that I wanted to wrap this up with is two elements. First, if you don't have your symbol on here and you want to, just send an email to Ulla and you can be on here if you sign the charter, which is just outlining the rules of engagement. And then please, if you have other dates, you can get on this upcoming meetings and events calendar here. And for those who haven't seen this, there was a Regulatory Education for Industry annual conference by the FDA. This was a 4-day conference happening last week. And as a last point is that we provide the link to all those recordings and July 21st was the devices track recording. There's also a devices and biologics track, so I think that's a great resource to have. And if you have additional dates for August, etc., I think Brandon, your dates will be included soon. But please just submit those. And in case, we have moved this a bit around, but for communication, so any of our official communication, the complete archive of everything that we've done is available through this news and events page communication here with the archive. So just in case that was there was a question about this. So with that, we do a point landing, unless there are some other comments, suggestions. Wonderful. Thank you all for participating and hope to see you in about a month.
Thank you. Wonderful.